Okay, well, welcome everybody to this week's edition of Micro Seminar. This morning we have Paul Carini from the University of Arizona, who's going to be talking to us a bit about leveraging microbial cultivation to do some really cool things in terms of ecology and evolution. I want to, before I introduce Paul, just remind everybody that the best way to ask questions actually is through the YouTube live chat window. We can see all of that, and it'll also be recorded, so don't put anything on there that you don't want other people to see later. But it also gives us a recording of the questions that come in, so if we miss you at the end, Paul can go back and answer those. If you don't want to interact that way, you can hit us on Twitter via the hashtag #USeminar or the handle @microseminar. Okay, so let's get back to business here. Paul did his bachelor's degree at the University of Wisconsin, Milwaukee, and a master's degree there as well under the advisement of Chuck Wimpy. I met Paul at Oregon State University when he was doing his PhD with Steve Giovannoni. He went on to do a postdoc with Alison Santoro at the Horn Point Labs, and then a second postdoc with Noah Fuhrer at the University of Colorado Boulder. And after that, he got the opportunity to set up his own shop at University of Arizona, where he's been since. And so without any further ado, I'm going to turn over to Paul and let him regale us with tales of microbes.
Awesome. Thanks, Cameron. I just want to take a minute to highlight Jen Biddle and Cameron. A couple years ago, they had this wild idea to do an online seminar, which at the time was kind of like, sure, you know, we can do that. And it was a good idea, but it was really a visionary sort of step into where we are now. And to have Dave Baltris and Lizzie Wilbanks and the folks from ISME now a part of this and supporting it, it's really fantastic. I took part 5 years ago. I kind of came across the video a couple days ago. That was young Paul. This is older Paul. So yeah, thanks for setting this up. I'm really happy to be a part of it. And then just for all of you that are out there dealing with the current situation, you know, if you know someone, a loved one that's got COVID or you've been directly impacted, you know, my heart goes out to you, and I'm sure all of our hearts go out to you. I think about you guys daily. Likewise, if you know someone that's in the medical field that's dealing with COVID patients, you know, I think about you guys as well. And thank you for that service and to us at this hard time. And it really, if you're just struggling in general with the upheaval of your life, you know, a lot of us are willing to chat with you. Doesn't have to be about how you're doing. It could be about science just to distract you. And hopefully that's what this seminar will be for you. So with that, I'll get into the topic of discussion today. We're going to talk a little bit about how our lab group uses cultures to try and answer questions in microbial ecology and evolution in soil ecosystems. As Cameron mentioned, I'm at the University of Arizona in the Department of Environmental Science. And if you have any interest in the things that you see today, please visit our website or follow me on Twitter. We have more detail there, as well as links to all of the papers you'll see today. During the talk, in the lower right-hand corner, you'll see QR codes. If you have your phone handy and you turn your camera on and hold your camera up to that QR code, it'll bring you directly to the paper that we're discussing on that slide, so you don't have to go searching later. You can pull it up on your phone in sort of an interactive sense. Before I get into the details, I have to figure out how to change my slides. Okay, there we go. I do want to make an announcement. We're hiring. We're going to be hiring. We're sort of on hold now for the COVID-19 ordeal. The university's got some strict guidelines about hiring, but as soon as we're resume lab activities, we'll be hiring a research technician in support of a new project looking at arsenic metabolism in gut microbes, mouse gut microbes specifically. So this position will be, we're looking for someone who can culture anaerobes and then identify which anaerobes that they cultured using molecular tools, and potentially even as much as sequencing their genomes and analyzing those genome sequences. You'll be interfacing with a new PhD student that will be hiring probably next fall, not this fall, as well as potential postdoc in a couple years' time in support of this big project. So keep us in mind. I'll post something on Twitter. The job is not posted yet. When it is posted, I'll make that publicly known. Keep us in mind. So before I get into the soil topics today, I want to thank a few people that have been integral to the work that you're going to see here. So all of the people here have had some part in what you'll see today. I just want to highlight a few. Joy Custer, she was our very first student in our lab. She started as an undergrad my first fall here at University of Arizona. She's since matriculated to a master's degree and will graduate in a couple weeks' time. She was really integral in a lot of the dilution to extinction experiments that we'll be talking about, as well as some methods development that hopefully will be published in the near future. Also, Ryan Bartlemy, he joined our lab as a postdoc about 1.5 years ago, and he gathered some of the data from a bunch of different undergrad researchers and really compiled it into a manuscript, which was our first paper published in M-Sphere earlier this year. So we're really grateful for his contributions. And Izzy Viney, she's currently a lab tech in our lab and will be picking up some of our JGI projects and as a master's student starting this fall. We're really excited to have her. When I was at the University of Colorado, Pat Marsden had a big role in the Relic DNA papers that I'll talk a little bit about today. He was an undergrad at Metropolitan State University in Denver and would commute up to do a lot of these Relic DNA experiments and really was integral in developing that technique. When I was at Colorado, I was supported by a series visiting postdoctoral fellowship. I highly recommend that for anyone who's looking for a postdoc in the coming years. Fantastic program, fantastic place to live. So please, if you're interested in that, apply for it. And then currently here at the University of Arizona, the work that you'll see today is supported by UA through startup funds and small seed grants. And we've leveraged that into some JGI awards. The first genomes of which we just got back last week, and I'm really excited about. We'll talk about those really briefly, but we have a whole bunch of other data that's sort of in limbo right now because of the COVID-19 shutdown that JGI will be helping us analyze. So I want to just start out by talking about soil in a very general context. Humans have sort of always had this intimate relationship with soil. We've sort of intuitively known that if we take care of the soil and different cultures take care of soil in different ways, that it will return bountiful food in the form of crops and bountiful materials for shelter and clothing. And now we use soil to produce compounds for bioenergy production as well. What we've sort of lacked as a species is the knowledge, for most of our evolution, is the knowledge of what gives soil this sort of vital quality. Why is it that soil is so life-giving? This started to change in the late 1800s when scientists started to realize that the cause of this vitality was actually soil microbes, fungi and bacteria that had important roles in plant health, as well as some chemical cycling. If we fast forward to today, we now know that in about 2.5 tablespoons of soil, so a fraction of what this person is holding, there are as many microbial cells as there are people on Earth. And in that same 2.5 tablespoons of soil, we know that there are as many microbial phylotypes as there are bird species on Earth. So these are tremendously diverse ecosystems. They harbor a lot of microbial biomass just below our feet. They're literally one of the easiest to sample habitats. You just could take a spoon from your kitchen and scoop a sample up, and there you have a sample, a sterile spoon. I guess you'd have to sterilize it first. Despite having studied microbes in soil for so long and knowing a lot about them, there's still a long way to go. It's still a great unknown. And this is really highlighted very nicely by a paper that my friends and colleagues at the University of Colorado published. The lead author on this, Manu, collected about 250 soil samples from around the world and did a fairly standard soil microbiome analysis on those samples. What they identified was that there were 25,000 phylotypes across all of those soils, underscoring this idea that these soils are very diverse ecosystems. But they noticed that consistently, about 500 phylotypes comprise the majority of this data. About 40% of the sequence data was comprised of these 500 phylotypes. And that's what's illustrated here in this phylogenetic tree. This is just a 16S rRNA gene amplicon tree. But what I'd like you to look at are these rings on the outside, because they give us some very important information. If there's a mark on this outer ring or this inner ring right here, that means we have a culture of that phylotype. It's been cultured somewhere in the history of microbiology. If there's a mark in this second ring here, that means that that culture is fairly well described. It's something we would call a type strain or a reference strain. And typically we have detailed substrate utilization patterns for these. We might have lipid profiles. And in many cases, we have much more information than that through transcriptomes and things like that. In the third ring here, if there's a mark there, that means we have a genome sequence available for that strain. And the outermost colored ring is an indication of their ecological preference or their ecological cluster as defined by these metrics right here. So what I'd like you to take away from this is if you sort of look at this and blur your eyes a little bit, about half of it is filled. We have really good representation in cultures and genomes for this sort of upper half maybe here. And for the lower half, we have very little understanding or very little information available. We don't have a culture. Since we don't have a culture, we can't characterize anything. We don't have a genome, metagenome, assembled genome, or otherwise. So we know essentially nothing about half of the abundant and ubiquitous soil microbes, apart from maybe their distribution and their environmental preference. This is really the impetus for me sort of switching from some of my marine work that I did for my PhD and early in my postdoc to terrestrial microbial ecology. There's a lot of opportunity here. So what we'd like to do is fill in some of this by culturing some of these and studying them. I'll give away the spoiler. We haven't cultured anything yet from this list that has not already been cultured. But that's okay. I don't have tenure yet. And when I get tenure, maybe we'll go after some of these things a little bit more aggressively. So how do we start to understand the function of these uncultured microbes down here? Well, the way that everybody's doing it, or many people are doing it today, is using molecular tools. And this is just a little review to bring everyone on the same page of what kind of molecular tools people are using. So the molecular community analysis involves taking a sample of soil here, and we extract DNA from that using a kit or some other protocols. Then we amplify genes, and these genes can be marker genes for phylogenetic purposes, something like a 16S ribosomal RNA gene, or they can be functional genes, like if you're interested in nitrogenase, you could target NIF-H or something like that. Our new sequencing approaches use massively parallel DNA sequencing to sequence a good sampling of that PCR product. And then from that, we can do things like reconstruct the taxonomic diversity of a sample and the community composition of a sample. So this approach is limited by a couple important things. First of all, we're only going to see things in this final output that we can amplify with the primers that we use. So divergent organisms won't amplify, and we won't see them in those analyses. And the other thing that we have to keep in mind when we look at this is that an organism is not a gene. So these single gene markers may be good at characterizing broad characteristics of a particular group or phylotype, but there may be a lot of important variation nested within that particular phylotype that is missed by this approach. And I think that that is what has driven sort of the new push towards metagenomics, is that we're trying to get an indication of what that diversity within a phylotype is. So metagenomics is simply taking this extracted DNA and sequencing everything assembling it and annotating it, and then reconstructing genomes, metagenome assembled genomes, and predicting pathways and potential physiologies. So there are a couple things that limit this, particularly in soil as well. So first of all, we talked about the high level of diversity in soil. This presents challenges because we have to sequence a lot more DNA to get a good coverage of some of these genomes and these pathways. And especially when it comes to rarer organisms, they're very hard to pick up in these data sets. We're also skewed sort of by the data sets that we're, the databases that we're comparing our data to. We can't annotate every gene very well. And even well annotated genes may have multiple functions that are important in the context of the ecology of the microbe. And we sort of obscure that a little bit by relying solely on molecular data. The other confounding factor, and one that I think is starting to emerge, is something that we published on a couple years ago, and that is that in soil, DNA is stable for a very long time. So when you take a soil sample and you extract all the DNA, you're not extracting just the DNA from the living organisms and the dormant organisms, but you're also extracting DNA from organisms that have been dead for some undefined amount of time. This is old literature. This has been known at least since the 90s, probably since the 80s, and maybe even the 70s, that DNA is very stable in soil for weeks to years. And until relatively recently, we haven't had a really good understanding of what effect that extracellular or relic DNA from dead cells has on molecular analyses of soil microbial communities. So for my second postdoc in Noah Fear's lab at the University of Colorado, we investigated this, developed some techniques based on propydium monoazide removal of extracellular DNA to quantify how much relic DNA was in soil and the effects that that would have on microbial community analyses. I'll point you to the paper for the details on that technique, which is down here. And I'll just quickly summarize the findings of that. I won't get into too much detail about this. Basically, we found that some soils have a lot of relic DNA. So we're looking at percent relic DNA in a soil sample on the left here across 31 different soils for prokaryotes on the upper panel and fungi on the lower panel. And we can see that some soils have a lot of relic DNA in them. In excess of 50%, 60, 70, 80, 90% in some examples, but that we captured the whole span of soil, the percent relic DNA in soils ranging from soils that have very little to soils that have a lot. And the patterns that we observe in prokaryotes and fungi are similar, suggesting that some physicochemical parameters are controlling how much relic DNA is retained in soil. And we tied this to some important differences in community structure. Two of the important aspects of this work that we wanted to follow up on were, number one, if you were to plot all of these sites in the same multidimensional space, the microbial community structure, they would all appear as independent points. In other words, all the microbial communities of all 31 samples were distinct. So that's maybe not surprising. These were samples collected to be a diverse set from across the United States. Whether or not you remove relic DNA, these points remained distinct. So there's some spatial component to the community separation that we wanted to follow up on at a smaller scale. In other words, is there a point at which the spatial differences coalesce into the same microbial community? The other aspect was sort of a theoretical aspect, and we wanted to understand whether or not microbial relic DNA from microbes that are dead obscured temporal signals in microbiome data sets. In other words, when a cell dies, if you can detect its DNA for some undefined amount of time, in theory, those dead microbes could be dampening temporal signals, temporal patterns in microbial communities. So we set out to design an experiment that addressed both a small scale spatial heterogeneity investigation and how relic DNA might affect that, those dynamics, as well as a temporal study of soil microbial communities, and whether or not relic DNA obscures some of those temporal signals. We chose 2 sites from this study. We did that because we knew that they were full of relic DNA, that they had plenty of relic DNA in them. We also knew that they were easy to access, relatively easy, kind of fun to hike to. And they were heavily instrumented as part of the Boulder Creek Critical Zone Observatory. So we started this experiment just as we were finishing the initial relic DNA paper, finishing writing it. We chose this site in the Boulder Creek Critical Zone. It's called Gordon Gulch. It consists of a north-facing slope that's heavily wooded by lodgepole pines, south-facing slope that's not as heavily wooded, has different pines, ponderosa pines, and a lot of interspersed grasses. Very contrasting ecosystems, and literally this is like a 7-minute hike from one site to another, maybe a little bit longer than that. On each site, on the south-facing slope and on the north-facing slope, we set up a 9x9 meter plot that was subdivided into 3x3 meter subplots, as described here. So there's one of these plots on the south-facing slope and one of those plots on the north-facing slope. At each time point, we'd hike in here and collect soil from each replicate plot here every 6 weeks for about 6 months. And that's a little bit of a lie, actually. We did this for a full year and had to throw half of the data away, or at least not included in the manuscript. It's not thrown away. It's actually publicly available. There was a significant batch effect. So we extracted these data sets in 6-month fragments. So the first 6 months were in DNA extraction number 1. The second 6 months were in DNA extraction number 2. And there was a significant effect in both the bacterial and the fungal data sets that was based on those DNA extractions. Whether it was differences in the kits, we're not sure. But we opted not to use the second half of the data set. I brought this up at the urging of someone that I was discussing this with last month, who said we need to be talking more about this because batch effects are real and we need to be devising ways and thinking about ways to analyze that. So I'm happy to answer questions about that after the talk if you're interested. So after we collected the soil, we treated a fraction of each set sample to remove relic DNA. We left another portion untreated, and then we characterized prokaryotic and fungal communities by sequencing. We also took a subsample from each soil and used it for extensive nutrient analyses, and we'll talk a little bit about that in a couple slides. These are just some pictures of the sites. Here's our north-facing slope, heavily wooded, some nice snowpack. Our south-facing slope here, much more sparsely vegetated with trees, and we have a lot of interspersed grasses. And then we have automated sensor data from the Critical Zone Observatory infrastructure that gives us real-time volumetric water content across the 2 slopes, and real-time temperature across the 2 slopes. And you can see that these have very different water delivery patterns, especially during snowmelt. Right here on the north-facing slope, we have snowpack much longer into the year. On the north-facing slope, very little snowpack, more of an even distribution of water delivery on the south-facing slope. And the north-facing slope similarly is a little bit cooler and a little bit less variable than the south-facing slope as well. So the take-home message from this study that was kind of surprising to me is that spatial variability in these 9x9 meter plots is persistent over time. We're going to talk about data from the south-facing slope. It's very clear that there's spatial heterogeneity on the south-facing slope. Very easy to see it in an MDS plot. So we'll be sharing that data, but the same lessons hold true for the north-facing slope as well. Again, here's a reminder of our plot design. What we're going to be looking at is MDS plots of different time points. So each point here is a time point, and they're colored by the replicate plot number, or the subplot number rather. We're looking at south-facing slope data. Here are the prokaryotes, here's the fungi, and this is total microbial community. So these are soils that are untreated to remove relic DNA. What I want to draw your attention to here is this cluster of the purple. I'm not sure how well this is going to come through. They're somewhat similar, but the purple, pink, and the aqua and the green color cluster together here in the prokaryotic microbial community, and those are distinct from the rest of the colors here. These four colors, we also see clustering in the fungal microbial community, and again, they're distinct from the remaining colors. Those four spots here correspond to plot 5, 6, 8, and 9. So there's some spatial reason that these four, that the microbial communities in these four plots are clustering together in these analyses. And we see it both in the fungal and the prokaryotic community. And I stood over the site. I mean, it's not obvious, and looked at it. You know, it's not like there was a tree there or, you know, an elk pooped on it or something. There was no clearly obvious reason as to why this is. It might be something very subtle, like a subtly different pH in these four plots. Maybe was an underground rock or something that was causing the pH to vary slightly. We don't know the reason for this variance, but there is this significant spatial heterogeneity across these. When we remove relic DNA, we see the same clusters grouping together, showing that spatial variability is the dominant source of variability across these plots at the meter scale, and that removing relic DNA does not really have that much of an effect on it. There's maybe a little bit of a homogenization between these two groups here, but there's still a very distinct spatial signal. So this spatial heterogeneity really created some problems for us when we wanted to analyze temporal signals, because we're really not looking at one unified site. We're looking at these spatially distinct sites. So we had to control for that in our statistical analyses. And when we did that, we found that we observed temporal variability, significant temporal variability, whether or not we removed relic DNA in these plots. But that temporal variability was less important than the spatial variability. We did, however, notice that when you remove relic DNA, we see an increased variability in some cases of increased temporal variability in some cases after we remove relic DNA, suggesting that removing relic DNA might be affecting the magnitude or the types of temporal patterns that we observe. We did not consistently observe that across both slopes, and we did not consistently observe it in prokaryotes or fungi. So this might be soil specific, it might be group of organisms specific, but in some cases there was this significant variability. So after we characterized the sources of variability from these sites, we wanted to look a little bit farther into what we could learn about the biology of the sites, or maybe the ecology of the sites, not the biology, based on whether there were groups of microbes that behaved similarly through time together. To do this, we analyzed our relic DNA removed samples with a program called local similarity analysis. This was a program that Jed Furman has used extensively and helped develop, analyzing plankton communities in aquatic ecosystems. To my knowledge, this is the first time it's been used in soil ecosystems. So we basically are looking at temporal correlations between individual phylotypes in our samples. And when we found a significant relationship, we included that into a network analysis. So this is, again, data from the south-facing slope here. Each little circle here, each node on this network is a phylotype. The ones that have bold ring around them are fungi. The ones that have very little ring around them or a light ring, those are bacteria. No archaea actually made these networks. There are stringency thresholds were not crossed for inclusion, were not met by by any of the archaea. If there's a line between any two sites or any two nodes here, that means that there was a significant temporal correlation, a significant and strong temporal correlation. The colors on these are actually meaningful. We analyze this network using a modularity analysis algorithm. And what modularity analysis does is it identifies groups of microbes, or in this clusters of nodes, I'm sorry. In this case, it's microbes, that are more densely connected to each other than they are across to other modules. So these are colored by a statistical analysis of these networks. So what do we use this for? Well, we extracted the relative abundance data from each of these modules and plotted them over time to get an idea of how these different modules are traversing through time in terms of how their relative abundances change over time. So what we're looking at here, this is the normalized relative abundance on the left axis over time here. These are the different sampling points. And we just extracted these normalized relative abundances from each color group and plotted them on a time plot here. So my favorite one, we're looking at module 7. We have our relative abundance starts off relatively high in the autumn months, November of 2015. And then as it gets cooler out, we see a decrease in relative abundance. And then as it gets warmer out, these particular taxa tend to prefer, they tend to grow in their relative abundance. We then extracted this data and modeled this pattern using our environmental variables that we measured. Using random forest modeling. So here we're looking at the relative importance of each of these variables and how it contributes to predicting this pattern. So if we again look at module 7 here, we see soil temperature and soil phosphorus are correlated, positively correlated with the distribution of these relative abundances over time. And salinity is negatively correlated as well. About 25% of this pattern can be explained by the variables that we measured. These gray ones here, that means that there was no significant contribution of those particular parameters to this given model right here. So this is when, I'm my own worst critic, and I remember looking at this network, and maybe you found yourself doing this too, and look at, you look at it really hard and you think, what the heck does this mean? And I sort of had this existential crisis of sorts, thinking like, well, how can you ever get at what this network is trying to tell you? Each one of these lines at a minimum is one hypothesis to test, right? How many lines are there on this network? And it's probably more than one hypothesis to test. It's probably dozens of hypotheses to test to get at why do these two microbes that are connected in time have a correlated behavior over time. This is when I really started to sort of change my philosophy. This coincided with me starting my own lab group. I really started to change sort of the philosophy of how I wanted to construct my lab and what kinds of questions I wanted to pursue. I don't think any one lab could ever disentangle this network. So maybe I started thinking that maybe I should be looking at things that we can disentangle and actually testing pretty clear-cut hypotheses from square one. So when we started our lab in 2017, I started it with the goal of having a very holistic view of soil microbiology that doesn't only focus on relating ecosystem characteristics to molecular analyses like I just described, but really has this central component of experimental microbiology from which we can link results from the lab through various, this is just an example of how we can link them, various techniques. We can link it to molecular analyses of the environments themselves. We can also study functional traits or metabolic rates in the lab and relate those to the actual ecosystem characteristics that we're measuring in the environment. So our lab group really tries to be inclusive of this experimental component. The problem with that is that most of the model systems and the model organisms that exist right now for soil microbial ecology are not universally applicable to all environments. So the microbes we find in Arizona are going to be different than the microbes we find in somewhere like Massachusetts or Michigan or even Florida. So we need to sort of develop our own model systems for our own ecosystems to a degree. And that unfortunately takes a lot of time. And that's what we've spent our first year or so, really our first 2 or 3 years doing, is culturing new microbes to use as model systems for our respective environments. Luckily, this seems to be a theme in microbiology right now, is that people are sort of coming around again to the idea that we need to culture and have these deep culture collections. These are just 3 examples of some of my favorite papers that have not only shown that we can culture a lot of microbes, but these groups are using these microbes to really disentangle important aspects of host microbe interactions in this instance. And what I'd like our lab to be able to do is to really expand the numbers and types of soil microbes that are cultured moving forward. So here's human gut, here's some Arabidopsis, rhizosphere, and mouse gut culture collections. And I'm really a fan of these papers and the papers that have used these culture collections to expand our knowledge of host microbe interactions. So our very first experiments were designed to try to modify some existing high throughput culturing techniques that have been very successful in aquatic systems to work with soil. So, you know, these aquatic people, they have it so easy, and I don't know that they realize it. They go out and collect a sample, stain it, and count it on a flow cytometer, and they have their experiment set up literally within a couple hours, and in some cases they can do it on the boat. For soil, it's much more difficult to do these types of experiments because we have soil, we have all this stuff, and the cells are embedded in this stuff: mineral matrix, organic matter, necromass. There's all of these different constituents that we really need to remove to be able to do these types of experiments correctly. So we've spent a lot of time working on optimizing those separation protocols to get cells off of the mineral constituents. It's not perfect, but it's pretty good. We don't get all the cells off. We can get a lot off, but not all of them. And the composition of the cells that come off of the soil particles is slightly different than the composition of the microbial communities in the soils themselves. But we're doing a better job on that. So we've developed some techniques to remove these cells. We count the cells with flow cytometry. They are actually very clean once we go through this whole separation process. Then dilute those cells into growth medium to a cell density of about 1 to 5 cells per mil. There's a whole art to developing growth media composition. We're not going to get into that today. We'll just say that it's dilute growth media for now. And then we incubate those for about a month and screen them with high throughput flow cytometry. Once we've identified positive wells, we array those positive cultures into 96 well plates and cryopreserve them. So we preserve them in 10% glycerol and pop them in the freezer before we identify them. Then we go ahead and try and identify or screen the cultures for properties of interest. This is our bottleneck, and I think it's the bottleneck in every high throughput approach for culturing. Very easy to get thousands of these, much harder to identify what's worth working with, what's worth following up on. and so on and so forth. So that's an area that is ripe for improvement for those of you that are interested in developing new protocols to screen. This would be a great area for you to research. So in aquatic microbiology, this approach here has been used to culture some of the most important microbes in the ocean and some of the most important microbes in freshwater, SAR11, some of the actinobacterial lineages in freshwater ecosystems. And the reason for that is that this approach here tends to favor the abundant taxa. And in many cases, the abundant taxa are these highly oligotrophic microbes that are very slow growing and susceptible to competition on Petri plates. The reason it favors it is that if there's a greater proportion of those cells in the cell extract or in the initial inoculum, they're more likely to land in a well. So did we actually culture the abundant taxa in our samples would be the next logical question to ask. And the answer is yes, we did. So when we collected these samples, we collect for cultivation. We sampled from a very relatively deep, 55 centimeter deep portion of soil from a soil profile. And when we sampled them, we collected samples so we could actually do a microbiome analysis of the whole depth profile. And that's the data I'm showing you here. So we're looking at depth in a soil pit and the relative abundance of, in this panel, 4 different OTUs that we cultured. So we matched our 16S sequences from our cultures to the microbiome data set. And what we found is that we cultured quite a few bradyrhizobium from this experiment. This was the most abundant OTU in all of the soil samples at all of the depths. So our approach worked in getting bradyrhizobium. We also cultured some mycobacterium. This was our 11th most abundant OTU in this experiment. And as you can see, their relative abundances increase with depth. So as carbon availability decreases, this mycobacterial OTU tends to become more abundant. We also cultured a bunch of nocardioides, again, also showing sort of this oligotrophic phenotype, where as you go down in soil depth, these increase in relative abundance, and some arthrobacters as well. For those of you that have looked at soil data sets, you've seen a lot of these things. We have some cultures existing of these things as well. So now that we have all of these cultures, what the heck do we do with them, right? We can only do so much with any given set of cultures. So what we're looking at is trying to understand the intersection of ecological attributes of these cells and the physiology of the cells, and maybe even the genomics of what confers those ecological attributes. So just 2 examples of the projects that we're working on now. We're looking at desiccation and rehydration tolerance in actinobacteria and how that may have evolved over time. 2 of those strains that we just talked about, the Arthrobacter and the Mycobacterium, are both actinobacteria, but their cell construction is very different. Just as a little reminder, Mycobacterium have a very complex cell wall configuration, much more complex than the average gram-positive microbe like an Arthrobacter, for example. We want to understand that in Arizona, desiccation tolerance is a very important stress for these microbes, and we'd like to understand whether or not differences in cell wall composition or differences in the metabolic activities or processes that go on in these cells may make them more or less adapted to this arid environment. So to test some of these ideas, we're doing transcriptomics and metabolomics on cultures that have been desiccated very slowly. So we've built this humidity chamber, is what we call it, the chamber, where we can control the relative humidity over time in the chamber by changing out different saturated salt solutions. So each of these little steps here is us changing the salt solution in this humidity chamber, and we can really tightly regulate the relative humidity of the cultures. And then when you put water back into this chamber, the relative humidity comes back up all the way to 100%. So we have two chambers, one which is always hydrated, one which is slowly stepped down, and we filter subsamples of each of these cultures onto filters, put them into this chamber, and then sample them at the beginning, in the middle, and at the end of desiccation, and then again after rehydration, to get an indication of two things. First of all, how are they regulating their genes during a slow, controlled desiccation? Many studies that have investigated desiccation don't do a slow, controlled desiccation, even though we know that in natural soils, rapid desiccation has different effects than slow, controlled desiccation. The other thing is that most studies don't look at the rehydration steps. So we're going to look at transcriptomes from these desiccated samples and rehydrated samples, but we're also going to look at the metabolic composition of the cells themselves. How are the cell walls changing? How are the internal metabolites, especially osmolytes, changing over this slow desiccation? And then what happens when you rehydrate the cell? I have this sort of pet idea that if you're accumulating osmolytes all the way down to this point, when you see water again, you have a fundamental problem because you're going to want to pop. It's sort of the opposite effect of desiccation. So I have this hypothesis that either those metabolites are being released into the environment very rapidly, or they're being consumed really rapidly, or maybe osmolytes are not actually even being produced. And there's a different strategy these cells take. So at this point, I don't know the answer. Unfortunately, all of this is on hold. The samples are ready to submit. But JGI is closed, we're closed. So hopefully soon we'll be able to get these samples to the JGI. The other project, and that I'll wrap up with real quickly, is we're investigating how genetic microdiversity affects the physiology of an abundant soil microbe. So this is another JGI project that we're working on. If we go back to this phylogenetic tree and we ask the question, well, which is the most abundant and ubiquitous soil microbe? It's Bradyrhizobium. So when we started seeing these cultures, I thought, we know so much about Bradyrhizobium, like who cares? Let's move on to the interesting stuff. But then I started seeing it, first of all, in this paper, this paper, but we started seeing it in all of our data sets, from the surface down to over a meter deep in the soil profile. Bradyrhizobium is dominant. It's the most abundant microbe by far. So this is when I started to ask the question, well, maybe this phylotype is really multiple phylotypes, or multiple ecotypes are nested underneath that. And the abundance of Bradyrhizobium is really the product of the 16S rRNA gene being a shitty marker for the gene, or a poor marker, I should say. Sorry about that. So we started culturing these, and we're actually really good at culturing Bradyrhizobium. They take a little bit of time to grow in the lab, a month or two sometimes, before we see colonies, but we have several hundred Bradyrhizobium cultures. This is out of date here. We had 90 a couple months ago. We have, I think, over 100 now. And they're identical at the 16S level to this globally abundant phylotype. So as part of our JGI proposal, we're sequencing the genomes of these Bradyrhizobium isolates from different depths and different sites here in southern Arizona. And I can tell you we've sequenced the ITS region in between the 16S rRNA gene and the 23S rRNA gene. And the ITS sequences, which can be a good indicator of genome level divergence, suggests that we see both site partitioning across different soil pits of Bradyrhizobium, as well as partitioning by depth in the soil profile. So we have surface clades, subsurface clades at 25 centimeters, and another clade at 50 centimeters. Sometimes we see a little bit of variability within a site, but the cross-site ITS sequences cluster very clearly. So we're sequencing those. Right now we're working that up, or actually I shouldn't say that, we're working up the cultures to harvest for sequencing. And then we're going to do two things with them. The first thing we're going to do is a core pan genome analysis to see which genes are partitioning across sites or depths. And then we're going to select a subset of those isolates to do some detailed physiological work where we look at transcriptomes and metabolomes of these cultures grown under different nitrogen availabilities. Remember, most folks think of bradyrhizobium as being involved in nitrogen fixation. That's not necessarily true. There are many isolates now that lack the nitrogenase enzyme complex. So we're going to investigate how those strains actually behave under different nitrogen availabilities from both a transcriptional level and a metabolic level, with a very keen interest on understanding how pan-genome content, so the genes that are not conserved across these different isolates, are involved in adapting to these different growth conditions. So we're very excited about that. We're actually working with Ben Tully at USC now to look at the core pan-genome analysis of existing bradyrhizobium isolates. And I would just say this, that there's a tremendous amount of diversity in this lineage from our preliminary analysis within a single OTU. For bradyrhizobium, it looks like the OTU is a little bit misleading in terms of being a coherent ecological group. So just to wrap up, you know, a lot of what we do and what we're working on in the direction that we're heading right now uses the cell as sort of the central unit of ecology. There was a paper a couple years ago by Karsten Zengler's group that really highlights this. Some of the key questions that we would like to pursue is how do cellular responses to environmental variability affect the use and production of soil organic matter? So which constituents are important to support growth, and which cells produce some of those constituents for other organisms? This sort of gets at this idea of metabolic cross-feeding to a degree at the microbial level, the microbe-microbe interactions. We also want to understand how the responses to environmental variability are modulated by the cell. How are they sensed, and how does the cell activate the responses, and on what time scales does that occur in the environment as well as in the lab? Which responses are evolutionarily conserved core responses? In other words, responses that are sort of predicated on this idea of a core genome, the core genome contents, and which responses come and go in the variable gene content or the pan-genome content. I don't think we have a good sense of that for soils, highly diverse ecosystems such as soils. And then finally, one thing we're really interested in, which functions can be attributed at the phylotype level? In other words, what can we say about a phylotype in terms of their physiology and their ecological importance in a given environment? And are these functions at that phylotype level actually ecologically important? So with that, I will be happy to take any questions. I've been in presenter mode, so I haven't seen the whatchamacallit, the YouTube link, but I'm happy to answer any questions. So thank you.
Great, thanks, Paul. You have a lot of questions. I saw the concurrent views hit 144 at some point in there, and we're sitting at 134 right now. So we'll see if we can get through a few of these, and then the rest I think we might have to see if Paul will answer offline. But right at the beginning, there were some questions concerning the archaea and why those weren't necessarily making your cutoffs for some of the microbial ecology work.
Okay, so I assume that the question is related to why they weren't in the network analysis.
Yeah, specifically, well, one person said, how about archaea? Another said, why did archaea not make it in the clustering? Is the archaeal community generally more stable?
Yeah, so the clustering on that, there were archaea that had significant correlations, but they weren't strong. So I believe for that network clustering, we had set a criteria of it had to be a Q value of 0.05 or something like that. So there may have been archaea that met that threshold, but then there's something called an LSA score, which is similar to maybe a Spearman correlation or something. And we had set that to be strong, so above 0.7. And the archaea didn't make that threshold of above 0.7. I will say that there are nitrifiers on the south-facing slope, and we nitrify both AOA-like organisms and nitrite oxidizers as well, but they just didn't make our thresholds. So they're there, and they're part of the ecology of those communities, and it's just one of these things, you pick a number, and if it's above it, again, it's kind of arbitrary. But that's just how it fell out.
Right on. By the way, if you want to drop out of presenter mode and just have your smiling face be the primary screen, you're welcome to do that.
Okay.
We have a question. Did the total number of microbes change in those experiments?
We don't know. Yeah,
Okay.
that would have been, so we had toyed with doing two things. So for the scale of that experiment, doing qPCR would have been really challenging to do. It was a pretty intensive project. So that would have been challenging. The other thing we had toyed with doing, but ultimately decided not to, is spiking in a non-soil organism's DNA at a known concentration and normalizing everything to that to try and get at absolute numbers. But we decided not to do that. I don't remember the exact rationale, but no. And that could be driving some of the patterns for sure.
Okay. Ashish Malik asks, what's your message from the Relic DNA work for the community? Does it only matter when studying fine temporal patterns?
That is just a fantastic question. And it's something that when I talk to people about this, I try and emphasize. It really depends what your questions are. Do I think that every study needs to remove relic DNA? The answer is emphatically no. I think that if relic DNA would obscure your conclusions in the end, that's something you should give thought to when you're designing your experiment. Spatial heterogeneity is dominant, and relic DNA has very little impact on spatial heterogeneity. So if you're just comparing soil 1 to soil 2, or it doesn't even have to be soil, it could be poop samples, it could be aquatic samples. Well, I guess it does have to be soil because maybe spatial heterogeneity is not important in those other ones. The spatial variability is going to be so dominant in the comparing those two samples that removing relic DNA is not going to change the community of one into the community of another. And that's true even at the meter scale. It may be true at finer scales, although we didn't address that in our study. And if you're going to do a temporal study, you have to, the most important thing to account for is the spatial variability. So in order to detect these subtle temporal signals, you have to sample a lot of space to have enough data to be able to disentangle that temporal signal from it. If you just sample 5 sites that are 5 meters apart over time, it's unlikely that you're going to see a temporal signal without sampling that intensively over time, if that makes sense. It's really, I think the relic DNA in terms of the temporal variability is to, if you want a really accurate picture of how the temporal variability is proceeding, it might be important to remove relic DNA from those samples. So.
Okay, very cool. Let's see, a few more. In terms of There are a number of questions on here that I think got answered actually in the talk, but you'll want to come and check this out in case there are a few things that you could potentially add some information for.
Okay.
Here's one: are actinobacteria known to target archaea with antibiotics?
Oh, I don't know.
Okay.
I don't know. That's a good one. I don't know.
Okay. Let's see, moving on to the growth screening experiments. Let's see, were the incubations, were they aerobic or anaerobic?
They were all aerobic. So we would love to do anaerobic, but the procedure itself is challenging to start with. And then you try and do it all in an anaerobic condition, and that becomes very difficult. And that's an advanced sort of manipulation for someone who's pretty experienced with doing these types of experiments.
Yeah.
So we would love to do that. We haven't done that yet.
Okay. There's a couple questions on the desiccation experiments. Was there, let's see, desiccation rehydration, what's the time scale of the treatment?
Yep. So we would love to mimic what we see here in the desert, which is usually a 3 to 5 month long dry period followed by very rapid rehydration. But I can't find people that want to wait that long to get their data. So we're looking at condensing that over about 2 weeks. So the dehydration happens. I think we change the salts every 48 hours. So each one of those steps is 2 days. And I think there's 8 or 9 steps. So about 2 weeks.
Great. Another person asked, how did desiccation affect growth?
We're looking into that in a so generally speaking, desiccation will slow growth as water availability decreases. What we're looking at trying to quantify somehow, but we don't have a great way of doing it right now, is to quantify how it affects viability over time. So what portion of those cells become non-viable over those 2 weeks? And then once we rehydrate the sample, how many of those cells maintain viability? The challenges we're facing with that is we can't find a great way to assess viability that matches up well with the way we're desiccating them. There's some technical reasons for that. But what we're probably going to end up doing is plate counts. Very old school. These all grow on Petri plates, where we take a filter out of the desiccation chamber and then plop it onto a Petri plate and try and get a count of how many viable cells there are on that chamber. But there are some technical reasons that that's a little challenging too, but hopefully we'll be able to overcome that.
Cool. Going back to the Relic DNA, there's a couple of technical questions. What effect does storage at -20 have on Relic DNA? And does the Relic DNA treatment continue after removal from, does the Relic DNA treatment continue after removal from -20 storage, causing you to lose some of your previously unlysed cells?
So the first part is we don't know what effect it has. Storage at -20, we don't store samples. We process them fresh. And I think that that's probably important, but I have no data to support that. And then what was the second part of that?
It might be irrelevant now if you're not storing them, but does the Relic DNA treatment continue after removal from -20 storage, causing you to lose some of your previously unlysed cells?
I don't think that matters. So the way that I don't know the answer to that. The way that we do this is we treat the samples to remove Relic DNA. So that's adding Propidium monoazide, and then we expose them to a very bright light. That bright light activates PMA. At that point, we consider the samples stable enough to freeze. So we freeze the samples at that point. And then when we do our DNA extractions, we thaw those samples that have already been treated and put that into our DNA extraction tube and do the DNA extractions as normal.
Okay, great. Okay, so some conceptual questions that I think you'll love. Is there any information from the growth screening step that can be used to support the abundance copiotroph / oligotrophy hypothesis?
I'm not sure what hypothesis. I guess I'm not sure what hypothesis specifically. So we're working on something right now about the whole concept of oligotrophy and what might be behind that. And I mean, there are definitely organisms that prefer high nutrients. There are definitely organisms that prefer low nutrients in their natural settings. But when we try and grow them in the lab, I think what we're trying to do inadvertently is just match their condition close enough so they don't die for some physiological reason. But yeah, we're looking into that. I think there are definitely oligotrophs in soil. We've cultured a bunch of oligotrophs that will only grow on low nutrient media. If you start ratcheting up the concentration of the nutrients they grow on fine at low concentrations, they are inhibited. So we're looking at some ideas and concepts behind that now. No experiments at this point because they're really hard to work with. But yeah, there's definitely oligotrophic taxa in soil.
Yeah. Okay. Let's see. I think we've got most of them here. There's one more big conceptual one. That is probably a good one to end on. How can we go about isolating auxotrophic microorganisms? Don't many uncultured organisms fall into this category?
My thoughts on this have changed. So strong convictions, loosely held, I think. My thoughts on this have changed. I'm not sure that unculturability is the result of strict auxotrophy as much anymore as maybe I previously thought. I think there are some aspects of that that might be true. But, you know, it's a complicated problem to solve. So the way to deal with auxotrophy would be to add a lot of stuff, right? Like, here's a diverse mix of compounds, eat what you want. The problem with that is that it's not like that in nature, where these cells are if, let's say, they're auxotrophic for alanine, and that alanine comes in through a very general amino acid transporter. And if you give cells a mixture of amino acids of which alanine is in there, they should grow. But what if glycine blocks that transport of alanine and there's twice as much glycine in your amino acid mix? They're not going to be getting that alanine that they want. They're not going to be able to grow for whatever reason. So I don't have a good answer for this, but I don't think that auxotrophy in and of itself is the reason we can't culture a lot of things. And genome data kind of supports that. A lot of these pathways appear to be complete. And oligotrophs in particular have a lot of transporters. And my guess is that the secret into culturability has something to do with how these cells are perceiving their environment through those transporters. And I don't know the answer, but that's my general thought on that. It's changed over time.
Cool. I scrolled down and I found a whole bunch more questions down here. Let's see. Roland Hotzenpigler asks about, he's wondering if your cultivation approach compares to results from heavy water studies like those of Bruce Handgate. Do you see similar taxa?
So
familiar with that?
I think you're referring to the studies that are showing which cells are active in the environment. So they use heavy water to ascertain growth and which cells are active. I haven't compared the data directly rather. But my hunch is that it's going to be roughly similar, and we would love to, we've got some ideas on how to use heavy water, especially with the desiccation studies to ascertain metabolic activity and things like that, but we haven't really pursued any of that yet.
Okay. Let's see, did pH or DO percentage change in those soils within the plots? Could this be explaining the major variance? I guess this goes back to your original, the initial data you showed.
pH or DO?
That's right.
Don't know about DO. We didn't measure I'm assuming that's dissolved oxygen. Is that DO?
Yeah, I would think.
Don't know about that. We generally assume that these are all aerobic, so they're surface soils. They're not compacted very much. So we generally assume that they're aerobic and that oxygen wouldn't be much of a factor unless you're looking at within a particle or something, but we didn't get to that level. And then the second part, pH. So the pH studies were done, or the pH that we measured was not on a per plot basis. It was, per subplot basis. It was on a per plot basis. So we don't have that fine resolution for pH, and that was due to sample quantity constraints. We couldn't collect enough sample at each time point without totally destroying the sites. So we had to scale back some things. So we don't know about pH. My hunch is that it's pH. That's my hunch, but we didn't measure that.
Okay, and there's one more technical question about why you set the flow cytometry threshold at 10 to the fourth cells per mil.
That flow cytometer can't detect cells at below 10 to the fourth cells per mil. So the way it's a Guava easy site flow cytometer. So it uses a capillary based approach to count the cells, where it actually measures how much liquid it's drawing up in through the capillary, and then it calculates the cell density based on that volume plus the number of events that occur. So the limit of detection on that system is about 10 to the fourth cells per mil. And that probably has to do with things like the physical parameters of the capillary itself. And you just don't see densities lower than that. It doesn't matter how long you run it. You really won't see densities much lower than that. So that was our clear threshold. You can also kind of start to see where a peak starts to form at 10 to the fourth. Anything lower than that, it's really kind of chasing ghosts, and we don't chase ghosts in our lab. So.
Awesome. Okay, well, I'll let you check out the comments. You got a lot of kudos, a lot of accolades from your colleagues. And if there's anybody that feels like we didn't get to your questions, you have something technical. There were some technical questions in here about the nitty gritty of how you do what you do, or why some of the data looked the way it did, that I think could be answered potentially via an email.
Okay.
That is a little bit more to the underlying, like, methodology. But if you feel like we didn't get your question to Paul over this, just go ahead and shoot him an email. He put his website and his Twitter all at the beginning of the talk, so you can scroll back to that. Thank you very much, Paul. And thanks for all the kind words at the beginning as well. That was wonderful.
Yep, no problem.
So with that, we will sign off, and there will be another micro seminar next week, so stay tuned.
Great. Thanks, Cam. Thanks, everyone.